Okay, welcome back. Dr. Lindner here. And just to go over a short review with you, again, I'm stalling a little bit only because I've noticed a few videos in the past had about a 30, 40 second delay. You're able to hear me, but it was blacked out. So just in case that is the case, I'm just going to talk for about 30 seconds and just remind you to go over some of the basic terms like what is a muscle fiber cell? What is the endomysium? What is the fascicle? What is a perimysium? What is the epimysium? What is fascia? What is a tendon? Where do tendons attach to bone? Right? All good review terminology. Don't forget to review the aponeurosis and what muscles do you have in aponeurosis. So now we'll talk about the NMJ, which is referred to as the neuromuscular junction. You heard me in a previous lecture talk about how nerves from the neck, from the brachial plexus, go to muscles of the upper extremity, and that nerves from the lower part of the spine, emanating from the lumbar plexus and sacral plexus, go to all the muscles of the lower extremity, the glutes, the iliopsoas, the quads, the hamstrings, the dorsiflexors of the foot, and the plantar flexors of the foot. Okay, if muscles are being stimulated by the neural system sending impulses, then those muscles contract and those proteins can thicken, and we call them hypertrophy. But if there's nerve damage to a muscle, if there is a severe subluxation in the neck, you can have atrophy of the biceps or atrophy of the triceps or atrophy of the muscles in the hand. I've had many patients over the years that have had carpal tunnel syndrome in the hand. And the severity resulted in atrophy of the thenar pad muscles. The thenar pad muscles are the muscles at the base of the thumb. The hypothenar pad muscles are the muscles at the base of the pinky. Okay, so they atrophy. So atrophy and hypertrophy are opposite terms. The muscle cells are going to be stimulated to contract by motor neurons. These motor neurons are called somatic motor neurons, where the somatic motor neuron and the muscle or the end plate of the muscle meet is referred to as the neuromuscular junction. And what we're looking at here is the NMJ, the neuromuscular junction. You see an axon on the top left here, and you can see that right here, it is myelinated. Okay, this is probably a Schwann cell that myelinated this axon. Remember, myelin is this fatty substance made of fat and cholesterol, and it speeds up the conduction of nerve impulses. It's really important to have that. Whenever myelin breaks down in an autoimmune condition, we refer to that as MS or multiple sclerosis. So this is the axon here. And now we have the axon branching off. We call these collaterals. We have all these different branches coming off. And at the very end of the axon is an axon terminal. And the axon terminal at the very end has this swollen tip. The swollen tip are called synaptic end bulbs. They look like light bulbs, so they're synaptic end bulbs. And what we have stored in these synaptic end bulbs, if we take this right here and we blow it up and enlarge it, we're looking at this right here. So this section, which is the synaptic end bulb, we see is here the synaptic end bulb. And what do we see in the synaptic end bulb? We see a bunch of these little tiny circles with this red substance in it, which is a protein that the body produced, and these are called neurotransmitters. Neurotransmitter is going to send a chemical message. Now, some neurotransmitters send a chemical message that are stimulatory, and some neurotransmitters are inhibitory. So some turn on the light switch and some turn off the light switch. The neurotransmitter that is involved at the neuromuscular junction is referred to as ACH, which stands for acetylcholine. Acetylcholine. And acetylcholine is always, always, always excitatory at the neuromuscular junction. Okay, so we see these vessels, and in the synaptic end bulb, we have neurotransmitters packaged in these little circles here, and that's acetylcholine. And this acetylcholine is going to move to the very end of the axon. And when it moves to the end of the axon terminal, it's going to open up. It's going to like fuse. It's going to fuse with the end of this axon and open up, which we see happening here. If we take this box and we blow it up, here we see the neurotransmitter being released through this method called exocytosis. Exocytosis is whenever it releases its substance outside of the vessel. Okay, we want to go back here to the upper right again and show you this axon terminal. We see lots of mitochondria there because it requires lots of energy, or ATP. We have these neurotransmitters, the acetylcholine, but something has to tell the acetylcholine to move to the end of the neuron or the end of the axon to release that neurotransmitter. And when it releases the neurotransmitter, it's going to be floating around in this gap right here in this space. The name of this gap or this space right here is referred to as a synaptic cleft. A synaptic cleft, C-L-E-F-T, a synaptic cleft. So this yellow gap right here is this yellow gap right here. This vesicle right here that's releasing the acetylcholine is this vessel right here releasing the acetylcholine. But what is it that's telling these neurotransmitter vesicles to be released? Something has to send a signal telling it to release. Well, that something is right here. It's called calcium. So calcium rushes in through a calcium channel, and it sends a message saying, hey, acetylcholine, do me a favor. We need you to move to the end of the axon, release yourself, release the neurotransmitter into the synaptic cleft. And guess what that neurotransmitter is going to do? That neurotransmitter is going to bind to this receptor. See right here, the green receptor. The receptor happens to be a sodium channel. So the ACH, or acetylcholine, is the key, and the key fits into this lock, and it says, open sesame. And when it opens, you have all of this sodium that rushes in to the muscle cells. Okay? And when sodium rushes in, it creates an action potential. For our purposes at this point, I know you haven't had neurology yet, but I want you to associate the term action potential with excitation, stimulation, or depolarization, all working together. So an action potential, excitation, depolarization, all working together. Okay? And I'm going to go through this a few times. This invagination right here, is called a T-tubule. And a T-tubule, the letter T, stands for transverse tubule. So here's another picture of a neuromuscular junction. These, again, are axon collaterals, and at the very, very end, we see an axon terminal. And way at the end is the synaptic end bulb. Okay, this is striated muscle, that is skeletal muscle. So when we look at it again, here, these are the muscle cells or the myofibrils. And look at these myofibrils. There's so many, and they're deep to this structure. The myofibrils are going to make up what is referred to as the functional unit of muscle called a sarcomere. A sarcomere is the functional unit of muscle. And a sarcomere is made up of different types of protein fibers. You may have heard of actin. You may have heard of myosin. Those are some main contractile proteins, but they're structural proteins. They're structural proteins and regulatory proteins as well, which I'll get into. But I also want you to see the mitochondria, all the mitochondria. For ATP production, muscles require energy. I also want you to pay attention to the T-tubule. I mentioned it before. This is a transverse tubule, and this transverse tubule runs transversely to everything else that you see here, like the blue mesh-like structure here, the spiderweb looking thing. That is called the sarcoplasmic reticulum. Now, the sarcoplasmic reticulum is super important. It has a major function. It's going to store a very important mineral. Guess what important mineral the sarcoplasmic reticulum will store? It will store calcium. Okay, the transverse tubule is where the excitatory action potential, nerve impulse, depolarization takes place. Okay, so let's see if we could put some of this together a little bit. Here is the somatic motor neuron. Here are the dendrites. Here's the nucleus and the cell body. Here is the axon. These are all little Schwann cells that are myelinating this axon. And at the very end is the axon terminal. At the end of the terminal are these synaptic end bulbs, and that's where the neurotransmitters are, referred to as acetylcholine. Okay, so if we take this box right here and we move it over here, this box has all of this going on. So this right here is the end bulb, and at the end of the end bulb are these neurotransmitters, those little vesicles. That neurotransmitter is ACH or acetylcholine. Something has to tell that acetylcholine to move to the end of the axon terminal and release itself into the synaptic cleft. The mineral that does that was calcium. Remember, calcium rushes in through the calcium channel, sends a signal, and all of a sudden we have this neurotransmitter acetylcholine being released through a method called exocytosis. Now the acetylcholine is going to bind to the end plate of the muscle. This is the end plate of the muscle. When acetylcholine binds, that channel opens and sodium rushes in. When sodium rushes in, we get this excitation, depolarization going down the invagination of the T-tubule. Okay? And this T-tubule, this transverse tubule here, butts up against the sarcoplasmic reticulum, which stores calcium. So here, the T-tubule butts up against the sarcoplasmic reticulum, which stores calcium, and the calcium will become released. When that calcium is released, it's going to sprinkle it to what's deep to it, which are all these myofibrils, that sarcomere, the actin and myosin, the thick and thin filaments. The thick and thin filaments create the striated effect. So we have thick and thin filaments here. The thick is myosin. The thin ones are actin. Actin is thin, okay? When the calcium interacts with this sarcomere, it can make it contract. In another video, I'm going to go into more detail on these steps that create this contraction and how it takes place. But for simplicity for now, we know that calcium rushes in, it tells the neurotransmitter to release itself through exocytosis in the synaptic cleft. That acetylcholine then binds to the sodium channel. The sodium channel opens, sodium rushes in, creating this excitation down the T-tubule, this depolarization. It butts up against the sarcoplasmic reticulum, releases calcium. Calcium is somehow going to make the sarcomere contract. In another video, I'll go into the magic that happens here. And then we have a muscle contraction, which I said is really just means muscle tension, okay? Now you heard me say that there's different types of proteins in here, right? Different types of muscle proteins. So some of them are contractile, like myosin and actin. This is what myosin looks like. These are myosin heads. And this is actin. These are going to interact together because they're contractile proteins. Now we have regulatory proteins called troponin and tropomyosin, and then we have structural proteins listed here, okay? If there is an autoimmune condition that attacks dystrophin, that's referred to as muscular dystrophy. Muscular dystrophy. This looks like a good place to take a break. When I come back, I will get into these proteins again, and we'll talk about the sarcomere and muscle contraction, how a muscle actually contracts.